Hello, my name is Michael Stürzel. I'm heading the division of molecular and experimental surgery at the University Medical Center in Erlangen. Sitting next to me are the two joint first authors of the work I would like to present to you. Dr. Elisabeth Naschberger, who is a group leader in my laboratory, and Andrea Liebl, who just finished her PhD thesis in my lab. Our work investigated the plasticity of tumor vessel endothelial cells in colorectal carcinoma. The hypothesis of our work was that isolated endothelial cells of colorectal cancers with different tumor microenvironments may exhibit different molecular and cell biologic features according to the tumor microenvironment they were originating from. The goals were to identify these differences, validate them at the tissue level, and to elucidate their specific function for tumorigenesis and the manifestation of the respective microenvironment. Based on previous work of our group, we decided to compare colorectal carcinomas with an interferon gamma-driven Th1 immune environment, which is associated with a better prognosis, with those tumors exhibiting a non-Th1 tumor microenvironment associated with a worse prognosis.
To this goal, we established an isolation procedure to obtain pure tumor endothelial cells from human colorectal carcinoma. With this technique, we isolated cells from 117 colorectal carcinoma patients and obtained pure tumor endothelial cell cultures in 58 cases. After careful quality control procedures addressing comparability of patient groups, tumors, the isolation process, cell age and integrity of the isolated nucleic acids, we obtained 8 endothelial cell cultures from the non-Th1 tumor microenvironment and 8 from the angiostatic Th1 tumor microenvironment. These cultures were compared by transcriptome analysis in the following. We found that tumor endothelial cell cultures in vitro obtained from the tumors with the different microenvironments maintain significant differences also in culture. The matricellular protein Spark-like-1 was the most differentially expressed gene, clearly upregulated in tumor endothelial cells from tumors with a Th1 tumor microenvironment as compared to tumors from the non-Th1 or control tumor microenvironment. The investigation of Spark-like-1 expression in vivo showed that Spark-like-1 is highly expressed and closely associated with endothelial cells in the normal colon tissues and colorectal carcinomas with a Th1 tumor microenvironment. However, it was significantly reduced in colorectal carcinomas with the non-Th1 or control tumor microenvironment. Spark-like-1 has been shown to be expressed in colorectal carcinoma already in previous studies, but expression of these genes was never detected in cultivated endothelial cells in vitro. Our in vivo results indicated that Spark-like-1 expression may be associated with quiescent endothelial cells as indicated by its expression in vessels of normal colon tissues.
To investigate this, we cultivated endothelial cells and kept them at confluency to induce quiescence for up to 10 days. Under these conditions, Spark-like-1 expression was induced as demonstrated by cytochemistry and Western blot analysis. Moreover, we could show that Spark-like-1 is also secreted from confluent endothelial cell cultures. We found that Spark-like-1 expression in cultivated endothelial cells is directly associated with the quiescence marker nuclear interleukin 33, inversely with the proliferation marker Ki67 and fully reversible, which are key postulates of cell quiescence. These findings confirmed that Spark-like-1 is associated with cell quiescence.
Validating these results again at the tissue level, we could confirm that Spark-like-1 expression is closely associated with major vessels with high pericyte coverage, low proliferation and increased vessel diameter, both in normal colon tissues and in colorectal carcinoma.
Moreover, we found that downregulation of Spark-like-1 expression in endothelial cell spheroid cultures increased angiogenic activity and that Spark-like-1 inhibited endothelial cell proliferation and migration and also sprouting in the spheroid. These results indicated that Spark-like 1 has anti-angiogenic activity and functionally may contribute to vessel quiescence. Interestingly, it has been shown by Hu and colleagues in 2012 in clinical cancer research that Spark-like 1 also can inhibit proliferation of colorectal carcinoma tumor cells. These results indicate that Spark-like 1 may be a vascular-derived antitumorigenic factor.
Finally, we wanted to know whether Spark-like 1 may induce vessel maturation or stabilizes mature vessels. To this goal, we investigated the impact on smooth muscle cell migration and proliferation, and we found that Spark-like 1 inhibits smooth muscle cell migration but does not have a significant effect on smooth muscle cell proliferation. This indicated that Spark-like 1 expression in the quiescent endothelium might stabilize mature vessels but might not attract further smooth muscle cells to increase maturation. In a Spark-like 1 knockout mouse model, we found that colon vessels show reduced vessel maturation and reduced vessel perimeters, similarly as observed in colorectal carcinoma.
In summary, these results led us to the conclusion that Spark-1 is a regulator of blood vessel homeostasis. It is expressed in quiescent, normal colon vessels, and this expression is still maintained in tumor vessel endothelial cells of tumors with the Th1 microenvironment. The inhibition of angiogenesis and tumor cell proliferation by Sparkle may contribute to the improved prognosis associated with tumors exhibiting a Th1 microenvironment. In tumors lacking a Th1 microenvironment, Sparkle1 expression is repressed and allows improved tumor cell proliferation and angiogenesis associated with worse prognosis. Interestingly, these differential microenvironments showed imprinting effects in tumor vessel endothelial cells, which are maintained in culture. At the translational level, the antitumorigenic activity of Sparkle may be worth to be considered in anti-angiogenic treatment approaches, which may foster progression of disease in tumors with a Th1 microenvironment. We have to thank numerous co-workers who significantly contributed to the success of these studies, and to several grant agencies which supported our work, specifically the German Cancer Aid, the German Research Foundation, the German Federal Ministry of Education and Research, and the Interdisciplinary Center for Clinical Research of the Clinical Center Erlangen. I thank you very much for your attention and interest. Goodbye.